Now I'm excited to introduce Ramarao Vepachedu, Development Scientist at Leidos Biomedical Research.
Good afternoon. Thank you very much for giving me this opportunity to present our work on CAR T cell production at Frederick National Laboratories. At Frederick National Laboratory for Cancer Research, there are multiple programs. These programs are designed to help advance the science and technology. Biopharmaceutical Development Program is one such program, and this BDP provides resources and expertise in development of investigational drug products. We support drug candidates from early development stage through manufacturing for phase 1 and phase 2 clinical trials. We work with multiple customers and funding agencies and have a broad area of focus. We support production of antibodies, recombinant proteins, vaccines, and viruses. We have a portfolio for gene therapy and cell therapy products. And in gene therapy, we work with the AAV. And for cell therapy, we are working with two, and we are manufacturing them at present. So the learning objectives for today's presentation is the CAR T cell drug product manufacture that we are doing. And we are trying to introduce new engineering technologies to get the CAR T cell production using CRISPR Cas9. So we'll be giving some introduction on that part, what we are doing. So the immune therapy is aimed to enhance our ability to fight cancer and pathogens. Chimeric antigen receptor T cell therapy basically aims to modify T cells to recognize the specific antigens on the cancer cell surface. In autologous CAR T cell therapy, patients' own T cells are isolated and engineered in vitro to express the artificial receptors that recognizes the cancer cell receptors as surface proteins and then give back them to the patient as a living drug. So since we are working with the live patient, the autologous CAR T cell production is between two areas. One is that from the patient, the samples has to be collected, and the manufacturing site, the manufacture has to be completed, and there should be a coordination. Basically, from the patient, the apheresis blood product is collected and cryopreserved, and then shipped to manufacturing site at Frederick National Laboratories. At Frederick National Laboratories and the GMP suites, we do the T cell isolation, T cell activation and engineering, and then expansion. Finally, we cryopreserve the CAR T cell product and then transfer it to the patient for infusion. So all these steps we do in a Prodigy bioreactor, and that allows us to do most of the steps like isolation to expansion in the closed form. So this is a bit more detailed how we do the autologous CAR T cell manufacture. In the first part, once we collect the blood samples, we get them in a cryopreserved form, and this apheresis product was thawed and washed, and then CD4, CD8 cells are enriched. That is basically we are collecting the T cells, and once the T cells are collected, they were activated, and we add cytokines also to induce the growth. Basically, at this stage, we are looking at about 100 million cells to start with. It's not necessary that all the time that can happen, but to start with 100 million cells is a good to go. Once we collect the cells and we start the growth, on day 1, we introduce the CAR T sequences into the cells. We use CAR lentivirus or CAR retrovirus. Either virus can do the job. We transduce the cells And the outcome of the transduction is shown in the inset. We have the T cells with the T cell receptor on the cell surface. And after the transduction, the T cells start expressing the chimeric antigen receptor on the cell surface. And then we allow the cells to grow for another 5-6 days. Basically, we allow them to go to the day 7. And then we wash the cells and make into the final formulation and control freeze the bags and send for the shipping. And we also do in-process sample testing, and we have a list of assays to be performed after the samples are ready for the shipping. And all these process steps are in general completed within 2 weeks because it has to go to the patient in as quickly as possible. So CAR T-cell therapy is very much successful, especially in B-cell lymphomas, and the success rate is very good, but there are major challenges with that. The major challenges for the CAR T-cell therapy is severe toxicities and exhaustion and suboptimal persistence. There are many ways to counter these things. First part in this case is the chimeric antigen receptor design itself is one of the major factors how the CAR T-cell therapy works. So there's a scientific community is working a lot on that one, and there are multiple generations of chimeric antigen receptor designs were made. We have generation 1 to generation 4 with various additions and efficiencies. But when it comes to the manufacturing, what are the things that we can actually do to improve the quality of the product? Gene modification with the newer technologies can be done at the manufacturing to enable us to produce more efficient CAR T products. We can do the knocking off specific genes to counter the exhaustion and suboptimal persistence. One other thing important for this, going for the new technologies, is when we use lentiviral retroviruses, these viruses generally once transduced, insert the sequences, CAR T-cell sequences, at random places. They are not very specific where they are going to introduce. And in many cases, they can go and insert at a functioning gene sequence. And these things are not a major problem, but it's a safety concern. And if we can actually deliver the CAR T sequences at a specific site, it would improve the safety of the CAR T-cell product. So how do we want to introduce the modifications? There are multiple technologies. There's recent publications have shown people have used zinc finger proteins, TALENs, and CRISPR-Cas9. And Dr. Marston's group has presented, Dr. Marston has presented in the morning how the CRISPR is doing a lot of improvement in the CAR T-cell production. So basically, we are using similar technologies. I would not go very deep into the explanation of how CRISPR-Cas works. I'll be defining basic things that are required for us to understand what we are doing here. Basically, there are two components, the guide RNA and the Cas9. The guide RNA is used to home in the RNP, ribonucleoprotein, to the site where we want the double-strand break to be introduced. So, in this case, when we use a specific site to introduce the double-strand breaks, the double-strand, once it break that happens, the cellular mechanisms start to repairing them. There are two major mechanisms that are involved in this. Non-homologous enjoining, where the cellular proteins tries to patch the double-strand break. And this mechanism is not so efficient in the sense it will sometimes introduce the insertion some additional nucleotides and or remove some nucleotides. thus causing insertions and deletions. If we introduce our double-strand break in exon region, these indels will result in frame shifts and knockout of the proteins. That's how we use the CRISPR-based mechanism to knock out specific genes that we are interested to knock out. At the same time, there is another mechanism, homology directed repair, HDR mechanism. In this one, the cell tries to find a homologous template and tries to repair the double-strand break. At that time, if we introduce a homology sequence containing our interested sequence inside, then the cell mechanism tries to copy that one and use that as a template and introduce the gene of interest that we have put in the homologous template into the DNA. We can use either double-strand DNA or single-strand DNA as a template to introduce our transgene into the cells. So the technology is there, but what genes we are looking at? We are producing the CAR T-cell therapy products. We are developing a platform that has the capacity to bring in the new technologies, and we should be able to do that one. So, but the genes that are interesting, that are targeted in these studies, are basically either T-cell receptor knockout. We have alpha chain and beta chain for the T-cell receptor. Since we are introducing a chimeric antigen receptor into the cells, we can take out the T-cell receptor and put the chimeric antigen receptor in that place. And the second set of the genes that we are looking at is immune checkpoint inhibitors. The CAR T-cell product, once it goes into the tumor microenvironment, will face a lot of hardships, and many a times there are inhibitory signals there. So if we remove this checkpoint inhibitors suppression, then the cells can perform better in the tumor environment. And the third set of genes that we are looking at is if we want to make the CAR T-cell product allogenic or universal CAR T product, we would be liking to take out the signal molecules that tell the host immune system that there is a foreign molecule there. So in that case, we will be trying to remove the T-cell receptor, and we can also knock out the MHC class 1 proteins, especially beta-2 microglobulin. So these are the target molecules that we can actually use to introduce the modifications in the CAR T-cell product. So at this time, we are more focused on, or we are working with track locus. We want to knock out the T-cell receptor and introduce that in the natural T-cell receptor, introduce the CAR in the natural T-cell receptor locus. So the blue highlighted area shows that T-cell receptor, and we want that to be knocked out, and we introduce CAR in that place. So we can achieve that by knocking out track locus and introduce in the same locus our chimeric antigen receptor. In addition, we can also knock out PD-1 locus. As I said, this one is will enhance the possibility of cells to survive in the microenvironment better. So what is that the modifications that we are bringing into the our platform? Our CAR T production platform is basically multiple steps are there. We are not going to change many of the steps. Only one step we are looking at to get the modifications to done. Once we get the patient samples, we isolate the T cells. And at the time we are introducing chimeric antigen receptor using either retrovirus or lentivirus. Now, instead of that one, we want to introduce sgRNA and Cas9, whose complex is ribonucleoprotein, into the cells. And at the same time, if we want to introduce the chimeric antigen receptor, we can put the HDR template along with it. And then continue the same process to produce the CRISPR-based CAR T cell product. So to go ahead and to do that one, we went and tested knockouts first and then knock-ins. So these are published in literature by multiple groups, and we know that these systems work there. But when we are bringing them into our platform, we are testing multiple sgRNAs and their efficiencies. So to knock out T cell receptor alpha chain, we can go ahead and introduce indels in track locus. So we designed and tested about 12 sgRNAs in the first exam of the track locus, and after 4 days electroporation of the sgRNA and Cas9, we tested what is the surface expression of the T cell receptor. As you can see in the last column, the knockout efficiency seems to be pretty good. Most of the sgRNAs are giving us more than 90% knockout efficiency. So for the track knockout, there are multiple sgRNAs that looks pretty good, and we tried the similar thing for the beta chain. The TRBC locus has two sequences, TRBC1 and TRBC2, and there's a lot of homology between these sequences. So we can design the sgRNAs for knocking out both the sequences or introducing the indels in both the sequences. So in here also we tested again at 12 sgRNAs, and the knockout efficiencies in this also seems to be pretty good, and we are getting many of the sgRNAs getting us more than 90% knockout. So the knockout using a sgRNA CRISPR-Cas9 mechanism seems to be pretty much working well in the track locus. And we multiplexed all the three, alpha chain, beta chain, and PD1, all the three together, and we multiplex knockout them and checked how the efficiency is working. The efficiency seems to be pretty much good even in the multiplexer knocking out. We are getting about 94%, above 90% for many of the samples that we have tested. And the PD1 knockout is also good. It is between 70 to 80% efficiency is there. So when on the surface flow cytometric studies, our knockout seems to be working pretty good. So what is that we wanted to do is that we wanted to confirm this at the genomic level also. The gene edited samples can be PCR amplified and send it for sequencing, Sanger sequencing, and use that Sanger sequencing to see the indels that are introduced by these sgRNAs. We have tested for all the samples, but I'm presenting one of the sample here. You can see that once the sequences come, we can analyze these sequences using multiple softwares that are available freely on internet. They are like DSD code is there, TIDE is there, and ICE analysis is there. We use ICE analysis here. So these analysis tells us that what are the indels that are introduced at the site of modification that we at the site of sgRNA target site, and we can see that that knockout efficiency, indel efficiency, also can be calculated using the software. So when we compare the indel frequency, we see this almost correlate with the numbers that we are seeing on the cell surface knockout. So the flow cytometric data as well as the genomic data is correlating pretty much very good. We are about 87–90% in the genomic assays. There are one or two modifications, they don't show up as much as we see in the cell surface, but many of the sgRNAs correlate with the cell surface knockout. So once we are sure that our gene knockout is working well, we also tested the gene knocking. For gene knock-in, we have used GFP and also CAR, chimeric antigen receptor sequences. We have used different lengths because in the published literature, they have used multiple sequences, multiple lengths, and we wanted to establish a protocol for us, and we tested multiple sequences. 300 to 800 base pairs homology arm length was used for the track locus. And we have used various amounts of DNA also. We used 0.5 micrograms to 6 micrograms of DNA per million T cells. And finally, the knock-in efficiency coming about 5 to 24%, which seems to be reasonably good. If we look at viral transduction that we do using lentivirus or retrovirus, we are about 40%. Especially in case of lentivirus, we get about 38% in our clinical products. And the published literature says that if we use AAV for gene delivery, the gene delivery is about 5 to 40%. And in the published literature, it is also shown that CRISPR knock-in using double strand and single strand, the range is between 1 to 40%. So at this point, we are getting about 5 to 24%. It also depends on the size of the chimeric antigen receptor that we are introducing. We will be introducing chimeric antigen receptors sometimes with promoter and sometimes without promoter. Those things will affect the size of the chimeric antigen receptor and then the outcomes of the knock-in efficiency also impacted by these size differences. So at this point, we are pretty much confident that, okay, we can knock out and knock in, and we can introduce them into the GMP process. We did most of our work at a small scale level. About 5 million cells is every sample study we used. Once we want to go for the scale up and in a clinical production run, the number of cells that we are using in a clinical production is about 100 million cells. So the amounts that we require for this is about 20 times than what we are using at a small scale. So now if we want to go to the GMP production, we need to know whether we can get them at a GMP ready condition, the amounts that we want to get it to go into the GMP production. In case of sgRNA, they are commercially available. mg quantities of sgRNA can be obtained, and they are compatible with the GMP requirements. The same is the case of Cas9. Cas9 can be obtained in milligram quantities to gram quantities, and it's a bacterial protein, so it's much easier to produce. So both the two reagents that are required, sgRNA and Cas9, are available and can be easily obtained for the GMP production. The HDI template is the one which we are looking at. The HDI template can be either plasmid or we can use single strand DNA. So there are possibilities that with the HDI template. In the HDI template, if we are using a plasmid, okay, that's pretty easier to get as a reagent for the GMP usage. But the plasmid has bacterial selections will be there, like a kanamycin or something will be there, sequences will be there, and the RE sequences also will be there, bacterial origin sequences are there. And the better way to is to avoid those sequences and only get the HDR sequence. To get that one, either we have to do the PCR from the plasmid, or we have to use restriction digestion to get the sequences. So that is a bit complicated process compared to the plasmid. When we come to the single-strand HDR template, there are multiple methods that are being used in the literature, like exonuclease digestion. There are kits available, we can use them, but the productivity of that is small. We will not be getting milligram levels of that one. So that's one of the things that we have to look at it. And there are other mechanisms, reverse synthesis using RNA as a template, or we can use asymmetric PCR. All these things might work well in the lab R&D conditions, but when we want to go for the GMP and at a scale that we want to use, it will be a bit difficult to get those things. Finally, when we are searching out, we found that there is a decent publication in 2019 from Mark Bathe's group at MIT. These people are able to produce single-stranded circular DNA from the bacteriophage. And they can produce about 1 to 2 milligrams per liter of the cell culture. So this seems to be a better option for us. In comparison, this looks like a plasmid is for the double strand, and a single strand is a bacteriophage circular ssDNA. Both of them will have certain amount of issues, like you will have the selection marker there, like either kanamycin or whatever that you put it as a bacterial selection, and also we will have the RE sequences in there. But despite that, there seems to be the better choices to go ahead and explore the possibility to do the GMP productions. So basically, with all these things, we were in a stage that we are looking to go ahead and run a at-scale production run in a prodigy bioreactor. Basically, we are not making a great modification from our lentivirus CAR T cell production platform. We keep all the steps as they are, except one step we modify on the day one. We will exchange the lentiviral transduction with CRISPR-Cas and HDR, CAR HDR template. The difference in this case is that in case of lentiviral transduction, we are introducing the lentivirus into the cultures. But when we want to do the CRISPR-Cas9, we need to introduce them during the by electroporation. So that requires some engineering modifications. Either we have to introduce the inline electroporation protocols for the prodigy system, or we have to do that in a different way. We can use G-Rex flasks, and we can produce everything in outside the bioreactor in G-Rex flask type of cultures. However, at this point of time, it seems we are at the stage we can bring in this new technology into our platform and do the CRISPR-based CAR T cell production in our platform. So we are ready to go ahead and start that one. So all this work we have done at Biopharmaceutical Development Program. This is at Frederick National Laboratories, and we work under the Leidos Pharmaceutical Leidos Biomedical Sciences, and we work in collaboration with the Biological Resource Branch and Immuno-Oncology Branch of NCI. And anybody interested in our work or our technology, they can approach the Biological Research Branch. They will provide more information on that. Thank you very much for listening to my work at Biopharmaceutical Development Program. If you have any questions, please go ahead and ask them. Thank you.
Hello, thank you, Ramaro, very much for the insightful talk. This is Claire from Just Good Marketing, and I'm on host the Q&A session for this talk. And now let's start with our first question. So the first question is, what are the quantities of HDR template you expect to use in a clinical scale production run?
So at this point, we haven't finally decided the qualities of the HDR template, but definitely we are looking at the purity of the sample and probably the sequencing of the HDR template. These are the primary criteria. We will be looking at it. We haven't yet decided the quality for the HDR template. But these are the sequence, fidelity of the sequence is the one major thing that we are looking at.
Sure, that definitely makes sense. So a couple more questions about HDR template. So which form of the DNA template you think is better to use for production?
The literature suggests that, okay, single-stranded DNA is better substrate. But looking at the GMP point of view, ease and ability to bring that thing into the system is more important. So from my point of view, both the double strand and single strand should be explored. It depends. If we can get single strand, I know that GenScript can provide a sufficient amount for our research work, but we are not sure if we want to go for GMP, the GMP quality single strand DNA will be available. And you know, the problem with this clinical studies is it's not one run that we are looking at. When we are going to plan for a clinical production, we are thinking about 20 patients to 50 patients. So my estimation at this point, if we are using 6 micrograms, something like 6 micrograms for a million cells, if we are going for 100 million cells, we are looking at 600 micrograms of HDR template for each round. So we are at the milligram level for each round of production. And if we are looking at a 50 patient clinical trial, then we are looking at 50 milligrams. And we have to do a lot of testing on that, and all those things are involved. So it's ease at which we can get it, but preference seems to be basically at this point single strand. So we are working to get different ways how to get the single strand DNA as a template.
Definitely, and just to echo what you said, production of single-strand large scale definitely is challenging. We know a lot of people are trying to do that, and at GenScript, we're developing our own methods that right now we're able to produce that in other large scale, like milligram level, to support clinical studies. So just something we're developing, working on right now. So if any customer are interested, feel free to send us an email anywhere, and we'll get back to you.
Yes, sure.
So, yeah, we're definitely looking forward to more collaboration with your lab as well. We've seen some good results, and we hope to help you to move that into later stages as soon as possible. And another question is, what is the single-strand DNA template size you used in your knock-in? Have you tried to add any other donors like double-strand DNA in your lab?
Yeah, we have used double-strand DNA also, and we didn't do a lot of work, but it seems as successful as a single-strand DNA. But regarding the size, we are looking at multiple sizes. At this point of time, if we are introducing, let's say, the promoter region also along with that CAR sequences, the size will increase, and we are looking at about 2.5 to 3 kb size. So that's not very big size. At this point, we are looking at one candidate, and we might have a different candidate at different sizes. But at this point, we are at that place, and seems to be working well with that at that range.
Cool. Another question: do you see any toxicity with different donor template size or input amount?
Yeah, I mean, there is toxicity. That's what happens when you use the double-strand DNA. There is an excess toxicity compared to the single-strand DNA. But as if you have listened in the morning in the presentation from the Marston's presentation, the toxicity is one of the things that can happen to the T cells when we are introducing these things. And even if there is certain amount of toxicity, if you can recover the rest of the cells and grow them well, that should be fine. But the preference is basically if you can avoid the toxicity, that is the best condition. And we are looking whether we can get less toxic condition, that is using the single strand, and we can go with that to the GMP production. That will be the most preferred situation for us.
Yeah, definitely. And going back to Dr. Merson's talk, actually this morning, I think he also showed data where with this Cas9 targeting size design on the ends of the template, he's able to achieve like more than 40–50% of the efficiency. And here data you also mentioned the one job of the single-strand DNA right now is compared to, for example, lentivirus. And now the efficiency is typically 50% or higher. And new design, do you see this to be a promise of moving, the field into the non-viral approach faster, or do you see any other challenges? Any comments you want to provide for, the data he showed this morning?
Well, I mean, not any specific comment on that one, but there is a design factor that is involved in the efficiency. We can, at this point, when we use the single-strand DNA, we are touching about 24–25%. We might be able to improve on that because we just started and exploring multiple ways to get this thing. And on our first rounds, we get 24%, and probably we introduce certain more modifications and optimize some of the conditions. Maybe we can achieve higher transcription efficiency, knock-in efficiencies. And as you said that there are new technologies that he was talking in the morning. Those things can probably improve the efficiencies, and we have to check that one.
Yeah, I think this is definitely a fast-growing field with publications from different labs. So we're glad to see, how people are moving in this field since we're providing templates for researchers like you guys, but also it's just exciting to see, how fast things evolve. So I guess we have one last question is, and one last question. So for knocking with different templates using CRISPR, single-strand DNA, you showed efficiency of about 25%. Do you see any correlation between the homology arm length with the knocking efficiency?
At this point, I'm not clearly able to say that one. We do see the differences, but at this point, I would not be really saying that, okay, the size is the one that is driving that thing, because we didn't do in a organized manner to say that, okay, this is what it is doing. But the preferred choice is to increase the size, because the scientific thought process then you can use as long as side arms 800 base pairs that we are checking. Maybe we can increase that size also. But at this point of time, I would say that 800 is the one we are preferred number that we are running now. We have to check if the other ones are better or bad.
That totally makes sense. And that's, I guess, all the questions we have for the session. Thank you again, Romero, for sharing your work with us and answering your questions. Another reminder that GC Summit Talks will be available on demand after today, and this session will end. Thank you all for joining, and please stay again on track topics to join the next talk you're interested in. Thank you.